All right, let's go ahead and get started. Thank you so much. Good morning, good afternoon, good evening, wherever you are in this world. We're very excited that you're giving us an hour of your time. And we have two wonderful speakers joining us today. So I hope you're settling in with your drink of choice for an hour of great science. So today we will have two presenters. The first will be a short introduction to 3D genomics from our Director of Research, Kristen Sicking, which will set the stage for our guest speaker, Argyris Papantonis, also known as Akis, who will talk about his lab's pioneering research on the spatial organization of the glioblastoma genomes, highlighting its impact on tumor heterogeneity and providing valuable insights for targeted therapies. Finally, we'll wrap up with a Q&A at the end. So please keep those questions coming for a lively discussion following the presentations. So with that, I'd like to introduce Dr. Kristen Sicking, Director of Research here at ARIMA. Kristen holds a PhD in biology and leads and manages ARIMA's clinical and discovery research program. Kristen, the floor is yours.
So welcome everyone, and thank you for joining us today. I'm really excited to be here to share some of the latest from ARIMA Genomics. So today I'm going to begin with an introduction to ARIMA's technology and how we're able to explore the 3D structure of the genome before touching on some of the ways that 3D genomics is being applied in cancer research. So I'll talk first about how we can use 3D genomics in concert with other omics approaches to explore the mechanisms of gene regulation. And secondly, we'll discuss how to wield 3D genomics data as a tool to identify structural variants such as gene fusions. And finally, I'll talk about how to integrate the identification of structural variants with information about how that perturbs gene regulation in the local environment around a structural variant. So let's jump right in. To understand the 3D genome, let's start with what we might be a little more familiar with, what we would call linear genomics or a one-gene genome. We can define linear genomics as sort of the process of figuring out the order of each of the 3 billion A's, G's, C's, and T's of the human genome in each of our different chromosomes. But of course, there is far more complexity to the genome than can be easily captured in this linear paradigm. What we know now is that DNA is not just randomly shoved into the nucleus of cells. There really is a defined pattern to how chromosomes are packaged into compartments, and this is what 3D genomics is allowing us to describe. 3D genomics goes beyond providing the genetic sequence information. Its real uniqueness lies in its ability to offer additional insights through the spatial dimension of the data. Using 3D genomics, we are essentially able to sequence the structure of the chromosome, enabling us to identify structural changes like gene fusions or indels. Moreover, we can use 3D genomics data to measure the interactions between promoters and enhancers, gaining valuable insights into gene regulation. Now, what I want to start with today is this gene regulatory aspect and talk about how 3D genomics is an important part of a multi-omics approach to better understand the mechanisms underlying gene regulation. Now, in a multi-omics approach, a wide array of tools are available to link the blueprint of the genomic sequence to the various epigenetic factors, such as chromatin state, non-coding sequence modifications, which in turn can exert influence on gene expression or protein abundance or the interactions between small molecules within a cell. And 3D genomics plays a crucial role here in the multi-omics toolkit by identifying the long-range genomic interactions that can orchestrate gene regulation, thereby helping to bridge this gap between the DNA sequence and the epigenomic factors that contribute to changes in gene regulation. ARIMA has developed a number of tools to facilitate investigation of the 3D genome. Our core technology underlying these tools is based on Hi-C, which stands for High Throughput Chromatin Conformation Capture, which was invented in 2009 in Joop Dekker's lab. And at ARIMA, we provide kits and services that allow researchers to query the 3D genome. The product on the left here is what I want to focus on primarily. That is our genome-wide Hi-C analysis, which, as the name implies, looks at the 3D structure of the entire genome. But ARIMA also offers different targeted approaches to achieve reduced representation of genomic research regions of interest, including high CHIP, which uses immunoprecipitation to target interactions of interest, as well as promoter capture high C and custom capture high C, which use sequence-based probe capture to pull down genes or regions of interest to see how those elements organize in 3D chromosomal space. The general workflow of ARIMA's high C technology begins with sample prep using our kits. ARIMA's kits work with a wide variety of sample types and input amounts, including cell lines, frozen tissue, and even FFPE tissue, which is very common for profiling of tumors in cancer research. From a given sample, ARIMA's chemistry preserves the spatial organization of chromosomes within the nucleus by cross-linking the interacting pieces of DNA together. That DNA is then digested, tagged, and proximal fragments are religated to preserve that spatial information prior to NGS library prep. ARIMA's user-friendly workflows are fully compatible with existing NGS library prep kits to easily prepare samples for Illumina short-read sequencing and provide data outputs that can be analyzed using ARIMA's and other NGS analysis pipelines. So now I want to give you a sense of how we actually visualize ARIMA Hi-C data. This is a heat map used by the T2T consortium to scaffold the complete human genome sequence. And what you're seeing here is each of the chromosomes lined up across the top and along the side. Now, each point in this heat map represents physical interactions between two regions of the genome. So the dark red points that you see along the diagonal indicates that there are a lot of these 3D interactions between those regions. And the light red or white indicates minimal 3D interaction. Now, as you can see, it is very common for chromosomes to form these 3D linkages within themselves, but at least in healthy cells, chromosomes rarely form these linkages with other chromosomes. So let's zoom in to look at these interactions in more detail. What we're looking at here is about a 700 kb region on chromosome 2. And to help demonstrate how we would interpret this matrix, let's place 4 points along the interaction matrix here. And what you'll probably notice first looking at this is this bright off-diagonal signal with a high frequency of interactions. And what this represents is that the DNA near the purple point and the DNA at the green point are looping together to give that 3D interaction. To view that another way, we can represent these 4 points in this way along a linear genome. And from the interaction data in the heat map, then we would expect the purple and the green to be physically brought together. And likewise, the gray and the blue are physically brought together. So we can infer then that the chromosome is organized into domains by these loops that have formed in these regions. So now let's take a look at an example of how Hi-C can be used to gain really important insights into the mechanisms of gene regulation or misregulation in cancer. This example was published a few years ago by Goettingen and colleagues, and they used Hi-C to look at healthy cells versus cancer cells in T-cell acute lymphoblastic leukemia patient. And here what we're looking at is a zoomed in view of the region near the MYC locus, which is indicated here to the left. And nearby is a known superenhancer region. In healthy T cells, we can see that MYC is contained in an interaction domain that is clearly defined here by this sort of triangle and is separate from the domain containing the superenhancer. In TAL, however, that boundary between those two neighboring domains is largely gone, and both the elements exist in a single large domain. Looking at some of the associated multiomics data, we can see that the CTCF binding site near the domain boundary has been lost. And there's also an increase in the H3K27 acetylation signal in the area surrounding the superenhancer. Together, these data suggest that the superenhancer has been turned on and brought into proximity to MYC, leading to a misregulation of the MYC gene, which we also see in the gene expression data. So this is a really nice example of how the 3D DNA organization uncovered by Hi-C informs how MYC is regulated in healthy cells, but also gives really important clues as to how it is misregulated in leukemia cells. Next, I'd like to shift gears to talk more about how we can use 3D genomics as a tool to identify structural variants, including gene fusions. And what really catalyzed our interest and investment into this application of 3D genomics was a collaboration with NYU Langone that began a couple of years ago. They had a pediatric patient, a 14-year-old girl with a stage 2 glioma. And the tumor that she had was resected and analyzed with DNA and RNA sequencing panels, but no driver mutation was found. So the patient underwent standard chemotherapy and monitoring. About 6 months later, however, this patient experienced a very rapid progression, and the tumor returned quite aggressively. So NYU again performed comprehensive DNA and RNA sequencing of the tumor and again failed to identify any driver mutations. So at that point, we performed a Hi-C analysis using ARIMA's Hi-C technology, which revealed a novel structural variant near the PDL1 gene, which encodes a protein that helps immune cells recognize normal, healthy cells in the body. In some cancers, high expression of PDL1 prevents immune cells from recognizing and targeting those cancer cells, allowing the tumor cells to proliferate. So since the Hi-C data implicated a possible role for PDL1 in this patient, our collaborators at NYU decided to look further into that result. And in fact, for this patient, the ARIMA Hi-C result was confirmed with immunohistochemical staining, which showed strong and diffuse PDL1 expression in the tumor cells. So with PDL1 expression confirmed, the patient was then started on immunotherapy with the drug pembrolizumab. And since that change in treatment, the patient has responded very well and her disease is now stable. So what's particularly exciting about this case to me is that by using 3D genomics, we were able to identify a novel structural variant that was missed by these other genomic tests, and which provided the key functional insights that enabled the medical team to prescribe the best treatment for the patient. So now let's take a step back for a second to understand how we're able to use 3D genomics to identify these structural variants in the genome. If we imagine that we sequence a normal cell, we might get a handful of reads covering a gene located on chromosome A and some covering a gene on chromosome B. If those genes happen to be fused in a cancer cell, then with linear genomics, you are relying on the sequenced reads that span that breakpoint to see evidence of the fusion in these two loci. Now in 3D genomics, as we've already seen, we capture information about the 3D structure of the genome. And one really clear characteristic of that structure is that chromosomes most often form these interactions within themselves, but only very rarely between different chromosomes. So in a normal cell, you expect to see a lot of interactions in the region surrounding gene A on chromosome A, and a lot of interactions in gene B on chromosome B, but not many interactions that are spanning that distance. However, when those genes are fused together, as in a cancer cell, now those genes exist on the same fused chromosome, and we should expect to see many reads spanning that breakpoint. So by using this approach, we are able to leverage the 3D information to essentially amplify the fusion signal to detect these gene fusions. And we are able to do so in a way that is agnostic to the partners that are actually involved in that fusion. Now, to show you what this signal looks like in real Hi-C data, on the left, I'm showing, again, a normal cell line. And we see, again, that the interactions are primarily within the same chromosomes. By contrast, in a cancer cell line, we can see a very distinctive off-diagonal signal. So each one of these squares here is highlighting a 3D linkage or structural variant between chromosomes that is not present in the normal sample. And you can see how that amplification of the fusion signal around a breakpoint results in this very clear pattern that we can easily detect to identify gene fusions and other structural variants. As we've developed this technology, we've done concordance studies to compare ARIMA's Hi-C approach to other gold standard methods for identifying gene fusions, such as RNA panels or FISH. And in all cases in which there are known fusions, we are also able to find that same fusion using Hi-C. This works in a wide variety of solid tumor and liquid tumor types. So we've demonstrated here that we can identify the same gene fusions as existing gold standard methods. But what about other cases like this patient we've already looked at, in which the gold standard tests have failed to find any driver mutations? So is this case that we started with a really unique case, or could 3D genomics be more broadly useful in driver negative cases? So driver negative cancers affect a huge proportion of patients. Almost 500,000 patients in the US alone have tumors with no druggable targets per year. So we've designed experiments to determine whether we could use 3D genomics to identify the druggable targets in cases that were classified as driver negative by other gold standard approaches. We've analyzed over 200 cases to date that were previously characterized with DNA and RNA sequencing and didn't have an identified drug target. In 70 of those cases, we have found with the REMAS technology a fusion event associated with a gene that is a target for an FDA-approved therapy already available today. In another 12 samples, we identified fusions associated with genes targeted by drugs in ongoing clinical trials. And in another 29 samples, we identified biomarkers with diagnostic or prognostic utility. So in total, we've identified biomarkers in 50% of the driver negative samples that could be clinically actionable. Although, as you can see here, the success rate varies depending on the tumor type. We've further tried to validate those results in as many samples as we can using IHC. And in the vast majority of those cases that we've tested, we are actually able to confirm their oncoprotein activity, just like we saw in the PDL1 example. So what we're seeing over and over again with these studies is that 3D genomics can really help cancer researchers in a few key areas. First, 3D genomics can help us detect known fusions that are identified by other gold standard methodologies. But 3D genomics also enables us to find key gene fusions and structural variants that are missed by those other technologies. 3D genomics has also really helped to unlock access to fusions from archived FFPE specimens, which can be very challenging to work with in other molecular assays. Now, in the last section today, I'd like to show one example of how we can integrate these structural variant analyses with the gene regulatory analyses using 3D genomics data. Now, here I'm showing the interaction landscape around a gene ETV1 on chromosome 7 in a normal prostate cell. And next to it is the interaction landscape on chromosome 14 in the neighborhood of another gene, MyPol1. And in the normal cells, these genes have very little interaction between them since they are on two separate chromosomes. Now, if we look in prostate cancer cells, here we see the interactions again on chromosome 7 and the interactions on chromosome 14. And in these cells, there is a fusion event between these two chromosomes. And we actually see the expected interactions between the two chromosomes on the rearranged allele. So in the interaction data here, we can see these looping structures denoted by the circles that are forming between the ETV1 promoter and other regions in an intron of MyPol1 in a region that is also marked by DNase hypersensitivity. So to prove that these regions were functional, the authors of this study removed the enhancer sequences using CRISPR-Cas9. And they were able to demonstrate a clear reduction of the looping interactions across that breakpoint, as well as a reduced expression of the ETV1 transcript. So as you can see, this is a really exciting case where we can measure directly how that structural rearrangement of the genome can affect the regulation of the genes surrounding that breakpoint. In conclusion, I hope I've helped convince you today that there is much more to the genome than just sequence, and that 3D genomics is a really important part of our multi-omics toolkit. So I've shown you today that 3D genomics is concordant with the current gold standard methods for detecting structural variants, particularly gene fusions, and shows a lot of promise for improving diagnoses in a clinical setting by detecting fusions missed by other technologies. Finally, 3D genomics provides unparalleled access to the sequence, structure, and the regulatory landscape of cancer genomes, revealing novel biomarkers and therapeutic targets. Finally, I'd like to thank the many collaborators who have provided us with samples for some of the studies that I've highlighted today. And thank you all for your attention. I'll turn it back over to Ibrahim to introduce Argyris.
Thank you, Kristen, for that wonderful and excellent presentation. I'd like to introduce Dr. Argyris Papantonis. Argyris studied biology at the University of Athens, where he also obtained his PhD following postdoctoral work with Peter Cook at the University of Oxford. He set up his lab at the Center for Molecular Medicine in Cologne, Germany. Since 2018, he has been a professor for Translational Epigenetics at the Medical Faculty of the University of Goettingen, Germany.
I want to start by thanking ARIMA as a whole for having me give this webinar today, and also Kristen specifically for making the introduction so much fitting to the stuff I want to talk about. And I'm going to start right away. So my lab is generally interested in this relationship between the structure and the function of the human genome. And a way by which one can study this relationship is by perturbing it. So how would you do that? You would choose a system by which this is inherently perturbed and see what the consequence might be. And we do this, for example, as of late, by looking at cells that have escaped the normal route of development, especially for proliferating cells, which is to eventually hit the barrier of senescence and stop dividing, and escape into malignancy. And this would, of course, give rise to a tumor entity and its progression and evolution over time. Now, as an introduction, the work I'm going to discuss today has been pre-printed. So if you scan this code or if you use this DOI, you can find it on the archive. And it's being almost single-handedly led on the analysis side by Ting Zi, who's a Humboldt postdoctoral fellow in my lab, and wouldn't have been able to happen if Roberto Pallini, who's our neuropathologist from Rome in Italy, had not been generally providing samples and funding for this. So I'm going to be talking about glioblastoma. This is a very common and most aggressive primary brain malignancy. It has, an incredibly poor survival prognosis, and essentially people that have a diagnosis for glioblastoma do not really have a therapeutic solution for them. So we will be using, for this study, we will be almost exclusively using stem cells derived from the tumors, the primary tumors of patients. These are known to be the most resistant compartment of the tumor to therapy, and also be the initiators of tumor recurrence. And what is known to me, for example, as a non-cancer biologist, the sort of hallmark of glioblastoma has always been its incredible transcriptional and genetic heterogeneity. Now, I will introduce our cohort. We have 28 patients, of which some have, from some we have primary tumor material, from others we have relapsed tumor material. For 3 of them, we have pairs of primary relapsed tumor from the same patient, and the tumors emerge at the same position in the brain, and this will become important later. And we also have a funny pair of samples here. They come from a single patient, but the glioblastoma stem cells are derived from 2 different compartments of the tumor, from the central region and the peripheral. So we're going to apply high C to all of them. And the idea, as Kristen already has described, is that you can sort of get 2 birds with 1 stone. So you have a coverage that is enough to give you whole genome coverage, so you can find structural variants and copy number variants for that matter. But you also get the 3D information by which things come together in potentially regulatory interactions in 3D space. And once we've done all our 28 high Cs and we just map them to the reference genome, so this is very important, you need to map to the reference genome, and there's a reason for it, although these people will have really individualized genomes, and we'll get to that. So you map to the reference genome, and then you simply ask, how similar are any two high-C datasets after mapping. And as an aside here, I want to say that, for example, if you have a decent resolution mouse stem cell high-C dataset and one from a neuron derived from that stem cell, they are pretty close together. They look quite a bit alike if you do these kind of a correlation plot. But here, I think you can all appreciate that there's huge divergence in these high-Cs. Even pairs of samples from the primary recurrent tumor, which you could see here marked 275 and 275b, they're closer than others, but they're really not very close together. Now, why is that? This is because genomes from malignant cells are usually riddled with structure and copy number variants that give rise to non-canonical, non-reference structures. And actually was the work from Feng Yue and the University of Chicago that has pioneered our understanding of this, because they have been able, his lab have been able to generate algorithms by which, using machine learning, you can almost automatically identify structural aberrations in these high-C maps. And this is usually my favorite example. Whenever you see this sort of butterfly signal, like this one here, on a high-C map, this signifies an inversion for which the breakpoints are, perpendicular to the sides of the butterfly. And of course, you can identify everything else, right? A deletion is very obvious. You have a large region without signal. You can find duplications of different kind, and so on and so forth. Now, another thing you can identify in these samples, you can find copy number variants and you can interpret them. And we validated that this is the case by, for one of these samples, we also have whole genome sequencing. And as you can appreciate, we can calculate at the same resolution, which is roughly 10 kb in our hands, we can calculate copy number variation exactly the same way that you would if you had whole genome sequencing. There's very little to be desired on this front. Now, I'm going to show you one example from one of these patients. So this is our line 457. And I think you can appreciate in this high-C map that you see all the different chromosomes one after the other, from 1 to chromosome X down there. And you have all this signal off the diagonal. So this is, for example, chromosome 3 here, chromosome 4, and so on and so forth. And Kristen showed you these already. These aberrations in the signal that are really strong as regards the signal intensity, they actually connect different chromosomes in trans, and they represent translocations. And if I make a Serdos plot like this, where you can also see now structural variants within a given chromosome, you can appreciate there's a translocation here, for example, between chromosomes 1 and 9, but there's also a duplication on chromosome 9, and there's an inversion here, and there are multiple deletions and so on and so forth. So there's lots of variants. And if you just repeat the experiment by regrowing the line and having an independent replica, you get exactly the same things with very little variance. Now, how does this look like for our cohort? We actually identify an unexpectedly large number of structural variants. So we have, in the worst case scenario, 26 or 27 of them. In the best case scenario, probably for the patient. And in the worst case scenario for the patient, more than 180 of them. And you can see the dispersion is, so this is a very uneven, pervasive, but non-recurrent distribution. So it's extremely heterogeneous. Inversions are the least represented structural variants, and there's very little recurrence between different patients. So heterogeneity is the key word here. And you can see this here. If we correlate the recurrence, if we try to find common structural variants, you can see that the actual Jacquard index, which means essentially the percentage of overlap of their occurrence, is very, very low. And you can see the highest, for example, are the two lines that are derived from two different parts of the same tumor, the peripheral and the central. And even here, you don't have a convergence that is higher than sort of 45%. Now, if you survey chromosomes and you start asking where would these structural variants occur, we see, and I'm going to start down here, that they very often are clustered. We all need to keep in mind that this is a selection process. So very often, most often, the clones that we get to see are highly selected in the course of evolution of the individual tumors. But here you can see all the structural variants being clustered, and you can see how this looks like on the high C map with multiple events of different intensities occurring together with a copy number increase. So these will not necessarily all occur on the same allele. We don't have this information yet. We don't have allele-specific information. And also, this is a population study, right? So you study essentially the average of many, many cells. But judging from the intensity of these signals, this concerns a large majority of alleles in the population. And you could ask for complex XVs or particular subtypes of them, where do they occur the most? So for example, if you want to talk about duplications, you see that they tend to be in regions where you have GC-rich content, so many genes, and they are in the A compartment, so the active part of the genome, and the expression is generally high there. So you tend to select for duplications that involve active genes, which makes absolute sense. If you want to talk about inversions, for example, there's no bias for expression, and they tend to happen almost equally in gene sparse and in gene dense regions, and so on and so forth. And you can also ask whether these happen closer or further away from topological domain boundaries. So these positions that actually insulate genes and their regulatory elements. And again, you can see that if you're talking about duplications, the breaks tend to be close to the boundary. If you talk about, for example, deletions, they tend to be away from the boundary, deletion of regulatory elements or a gene that sits in the middle of one of these domains. So there's very rich information that you get about the relative position and the contribution of these structural variants to 3D genome organization and its regulatory role. Now, what one could do, and this has been done for other types of hematological cancers, you can actually take the gene expression subtypes of these types of cancer, and there are 3 well-known signatures for a glioblastoma, the classical, the mesenchymal, and the proneural. And what you can do, maybe I should change this, this will be easier. So what you can do is you can actually do RNA sequencing of these tumors. You can classify them in the different categories. And after you do that, we can actually classify 22 out of the 28, not all of them. You can ask whether features, high resolution features of your high C can explain the transcriptional subtypes. And to our surprise, that didn't work very well. So for example, you have some of the mesenchymal here clustering, sorry, these are the classical ones. The classical ones here clustering together, but some other just cluster away. And if you look at even high resolution features, they really get mingled up. So the heterogeneity is such that you can't have a clear picture. So what is going on? This is what we were trying to understand. So what we found is that these structural variants, they provide opportunities to chromatin to form potentially regulatory loops between different parts of the genome. And there are 2 pretty large surprises here. So I'll give you an example. So this is the HLA locus. You have 3 active HLA genes, and you have a deletion in between these 2, the G and the J. And very often, in actually 5 out of our 28 patients, you actually have the formation of a neoloop. You can see one example here, another one there. You can see the loop forming there, connecting the promoters of these 2 genes and activating them both. Now, if I look at neoloop formation throughout the cohort, you can see that some have really high numbers of neoloops forming, more than 1,000, and some have very few, in the order of few tens or hundreds. And what is fascinating is that all of the opportunity arises in hundreds of cases in all of them because of hundreds of structural variants that we can map. The existence, the emergence of a structural variant does not necessarily mean that a loop will form. So you also need compatible, in some manner, regulatory elements and genes on either side, for example, of a deletion, so that they can come together. So there is scope of mapping these. And one part of the scope is that you can uncover new predictive markers. And this is the HLA-G gene that I was talking about, which has until today not been associated with glioblastoma, has been linked to some extent with melanoma. So you can take this gene and ask in the TCGA cohort of hundreds of glioblastoma patients, how does it look like? Well, it is on average overexpressed over normal tissue. This overexpression is not because of copy number variation. You can see the copy number variation here between the high and the low patients, but patients that have a high expression of this gene, presumably because of this neoloop forming, actually have a poorer survival prediction. And overall, we have more than 6,000 of these neoloops to look into. Another type of neoloop would be the obvious one, connecting the promoter of a gene like IFIT, shown here, with enhancer elements in its vicinity. So here what we have is we have an inversion that brings these enhancers in proximity to the promoter and allows the formation of these two neoloops that turn on the expression of this gene, specifically in a couple of these patients, but not in the other ones that do not have such a structural variant. So this means that very often what Kristen presented as enhancer hijacking can actually take place. So the question is, how informative is this, right? How often does it occur? And we can actually see that the fraction of loops that explain such events is really higher for neoloops than for normal loops pre-existing in these genomes, or for enhancer loops that will eventually, even if you don't map that, of course, talk to a particular gene, which is not the case, for example, for promoter loops. And this, most of the times, is linked to a hyperactivation of these genes. I should have a small parenthesis here and say that there's been a very nice work from Feng Yue published a few months ago, where some of these loops can actually be classified as enhancer repressor loops. So instead of turning on a gene, which is what you would normally expect, the classical enhancer hijacking paradigm, you can have a repressor hijacking to turn off genes in the vicinity of these structural variants. Now I'm going to give you another example. Here's the ADAM9 gene, again, never really associated to glioblastoma biology. You can form, there's neoloops forming in its vicinity, two of them with a promoter, one that sort of encompasses and forms a new gene locus. This is because of the deletion here. And you can see that, again, if we go to the TCGA data, ADAM9 can be found significantly overexpressed in the cohort. It's not because of copy number variation. And you can actually see this poorer survival also being associated with it. So the point I'm trying to make here is that these neoloops that one cannot assess by having linear type of genomics, like whole genome sequencing, can be important. You can see here GoTerm analysis of genes linked to neoloops that are either enhancer promoter or promoter. And you can clearly see, for example, that glioblastoma signaling is significantly in genes relevant for glioblastoma signaling are significantly enriched in this particular category. And what I'm trying to get to is that neoloops should be co-considered when we're trying to understand the biology of these tumors. And I'll give you two very obvious examples. So one is CDKN2A. This is the famous P21 protein. Very often, these locus is deleted because this allows cells to escape senescence and cell cycle arrest. And you can see that in most of our patients, there are actually 13 behind this color here. We have a deletion in the P21 locus, but we have a neoloop formation in the P21 locus in 4 cases. And in these 4 cases, this also leads to the repression of P21, presumably via repressor promoter of P21 interaction. Now, another hallmark gene is EGFR. So many of these tumors are driven by overexpressed EGFR. And you can see here that the amplification of this locus is also a very highly occurring event. In our cohort, we have 9 GSC lines that are amplified for EGFR. But we also have another 4 that are actually, sorry, this was 3 here, and it's 4 here. We have another 4 that actually have an enhancer promoter contact that forms de novo, so a neoloop because of a structure variant that can turn on EGFR. This is all nice and good, and it's good for the biology understanding of these tumors and generally the evolution of the tumors. But are there chances that one can use Hi-C diagnostically to inform therapy, like in the example that Kristen described previously? Now, we have 1 patient out of the 28 that showed really high expression of MYC. And we looked into this patient. So this is the glioblastoma stem cell line 148. And what we found is that there's a translocation between chromosome 12 and chromosome 8, linking the MYC locus, a very small region, with a 1 megabase region carrying multiple really highly expressed enhancers. And you can see the formation of two rather specific neoloops. Not everything contacts the promoter. So these enhancers here and that enhancer there make a neoloop with MYC. And actually, to make this even more interesting, this whole translocation is then amplified, presumably as extra chromosomal DNA, in this particular patient about 30 times. This means that we have a huge overexpression in just a single patient of MYC, a gene that is, of course, a known driver of carcinogenesis, but not often involved in these types of tumors. But nowadays, there are inhibitors that one can use to target MYC, and there's one, EN4, which is a very recent development and can actually interfere with the phase separation properties of this particular transcription factor. So we can hit our patient that has the MYC amplification and translocation with this inhibitor, or a patient that doesn't carry that. So 62 here, and you can see the difference in how we are reducing the amount of positive MYC positive cells here. We have a significant effect in the ability of these cells to keep proliferating, and we have some increase in the amount of cells that are led to cell death. So we wanted to explore this a bit more. And we thought, is there a way of in silico modeling this interaction and trying to understand it? So what is the minimal? And I'm not going to go into excruciating detail here, but there was a very nice interface for modeling chromatin folding in 3D and structural variants by Mario Nicodemi's group a couple of years ago. And this is called Prisma. The problem with this for us was that this was not designed to find to make predictions and modeling in trans, so for a translocation, but only things that happen in cis, which are slightly easier to model. So Mariano, a physicist in our group, he actually took the task of making this work. And what he managed to do is not only make it work, but also make it have some predictive value. So again, I will not go into the details of the physical modeling here, but what we can have is we can have models that nicely recapitulate the 3D interaction that we see in our high-C data. They do this just by using two pieces of information, the K27 acetylation and the RNA-seq that we have for this one locus. We have as a negative control another locus where the translocation doesn't exist. We artificially fuse it, and as you can see, we cannot get neoloops here because we don't have the propensity heretofore. And then you can actually look at the simulations behind these reconstructed 3D maps from the modeling. And each one of these is essentially a single allele, or if you want, a single cell. And what Mariano found in this is that the interactions of MYC with any of the two neoloops, or with any of the two enhancers, is actually mutually exclusive. He could never find a situation. So here is MYC on chromosome 8, white, in this hub here with, you know, the large enhancer, the really broad one. And here it is chromosome 8 again in white, chromosome 12 in black. They come together, and you can see here the single enhancer in light blue. And this was, you know, a bit surprising. So Mariano took to the task of treating this as a single cell experiment and predicting the outcome. And you can see here in panel F what it would look like if we were to plot it like a single cell experiment. Very often in gray, you wouldn't have a contact because, of course, that's part of the game. But very often you would have this mutually exclusive contacts with either one or the other enhancer. And there are now, you know, well-established ways of making prediction of how the gene will burst into transcription on the basis of these contacts. And I think you can appreciate that there's very different patterns occurring in different simulations, which could actually explain the vast transcriptional heterogeneity for making this particular case in this type of tumor. And now, as a last bit, I would like to discuss this comparison between the primary and the relapsed tumor. As I said at the beginning, we have three pairs where we have derived glioblastoma stem cells from the primary and the relapsed tumor from the same patient. And these relapses were always occurred at the same position inside the brain. And what we find is, and this is not unexpected, of course, is that we have more structural variants in the relapse situation. This makes absolute sense because, of course, these patients have undergone heavy chemotherapy and radiotherapy. They've all undergone the exact same therapy, by the way. And of course, this will induce lots of DNA damage, presumably, and provide more opportunities for structural variants to arise. However, the prevailing theory that local recurrence of glioblastoma is essentially a continuation of the previous tumor cannot be justified by our data. And I'll give you a very simple example. You can see, for example, here, there are some amplifications that you don't find in the relapsed tumor. And let's say that this extrachromosomal DNA might get lost. There are here 3 very apparent strong deletions, 2 of which completely disappear from this data. It is impossible to lose a deletion and recreate the DNA that you've lost. So these and other such type of observations when comparing the 2 landscapes using Hi-C allowed us to predict that these are likely, they have a common ancestor, most likely, and it's difficult to assess that, but clearly they are not one the evolution of the other. The most likely scenario is that the primary tumor is removed by the initial wave of chemo and radiotherapy, and this gives an opportunity to the relapsed tumor to develop under the pressure of chemo and radiotherapy and provide a rather different new clone. And of course, if you look at the gene expression profile, the difference between the 2, you can find genes that explain the transition and the aggressiveness of the relapsed tumor. And then if you use the Hi-C, you can find that enhancer promoter contacts actually justify in all 3 pairs the majority of differentially expressed genes. And of them, the gain of enhancer promoter neoloops is what seems to make the difference for the activation of these genes. So I would like to stop here and conclude sort of 4 basic things that is at least our take-home message from this study, which is the first such large-scale cancer study in my group. So we can leverage Hi-C to generate patient-specific scaffolds of chromosomes and interactions. And when we do this in glioblastoma, we discover remarkable heterogeneity, both as regards structural and copy number variants. And what is really astonishing to me is that there's very little recurrence of most of these, the vast majority of these. The opportunity that these structural variants and copy number variants provide is that they allow patient chromosomes to give rise to neodomains and neoloops that seem to be specific for the most part for individual patients. And these can now not only explain transcriptional heterogeneity, but they can actually be used to uncover new dependencies over the course of time. So this is more or less it. I'm happy to discuss any aspect of this and also give you more details about the analysis in the back. And thanks again for taking the time to listen to me.
Thank you, Argyris, for that excellent presentation. And with that, I wanted to open the floor to questions. And I see that we already have a few in the box right now. The way I'm going to do this is maybe try to prioritize by those that are upvoted, but we'll try to get to as many as we can. And then, so the first one I see here is, can you explain the mechanism of neoloop formation and their connection to cancer?
Yeah, well, I think what is more interesting is when they don't form, but I'm happy. So you can imagine that the reason why the 3D genome organization is interesting is because, of course, you can make 3D domains that are to some extent insulated from their environment. So if you position a gene and its regulatory element inside this domain, the 2 are more likely to interact compared to if they were at very different domains, right? And now you can imagine that you have 2 neighboring domains, each one insulated to the other. So these are 3D chromatin entities that have some sort of a boundary between them, and we can discuss how the boundary forms. Now, if I create a deletion that will eliminate the boundary, the 2 domains will fuse. And now regulatory elements and genes that are not supposed to really see one another are found in the same domain. And to the extent that these 2 can be compatible, and this is a very long discussion, the enhancers can turn on, so the regulatory elements can turn on or off the gene that they find. So the formation of a neoloop, which is an opportunistic event, could actually give rise to a new regulatory event, either the overexpression or the suppression of expression of a given gene. So this is the sort of gist of it.
Thank you. And I guess a quick follow-up to that, are neoloops always caused initially by a structural variant, and then that structural variant then results in a new regulatory interaction that gets formed?
So, no. Actually, my big surprise in the data, so yes, this is true, of course, the statement stands, but my big surprise from the data was that not every time that you have an SV, do you have the formation of a new interaction, or do we capture the formation of a new interaction? Because of course there might be some that due to resolution reasons or whatever, we're not capturing. So indeed, we don't find a direct correlation between having many SVs and having as many neoloops. And this was a big surprise. And I think this comes back to this opportunistic event. There are going to be times where the structure variant is in a region that has really no effect. It could be in a gene desert and it's co-inherited with another structure variant that is meaningful for the evolution of the tumor. So that is one scenario. And the other one is that you do make the structure variant, you do bring things in the same domain or close by in 3D space, but they're not compatible enough to actually make a strong and meaningful interaction. And there are also in development, for example, lots of examples of these non-compatible or non-doable interactions. So the two, one doesn't necessarily mean that the other will happen.
Thank you. The next question I see here is, you said this is from the stem cells in the tumors. Were they flow sorted or cultured? And how does this change the DNA structures? Did you ever look at the stem cells versus a frozen or paraffin embedded tumor?
This is a very good question, and we would like to thank reviewer too for the question. So this is very fair. The way that you do this is you take the tumor, and while it's still fresh, you essentially embed it in medium and you grow out the stem cells. And there are well-established methods to do that. And they will grow out and lots of other cells in the tumor will die. So you do select for some sort of a subset of the tumor. Now, the way that this happens is it needs to happen relatively fast, and you would need to work with these at relatively early passage so as not to allow further selection. And this is what we've tried to do here. What we're doing now is we're actually, for very few of these, we have a bit of the tumor left. In ARIMA, they're good enough and they can now do FFPE samples. We have not really tried that. We're not brave enough, I guess, but we have flash frozen samples where we're now trying to isolate all the cancer cells from the tumor and just go and do a high C on them and see how close we're going to be to what we see in the stem cell compartment. They will not necessarily be exactly the same.
I see. Thank you. The next question I see here is that it looks like to me that EN4, the anti-MYC drug, also had an effect on the control clone without MYC neoloops. Am I right? It was fast. Yeah,
it wasn't deliberately fast. Sorry. No, it hasn't got an effect. And actually, we have tested 3 different non-MYC expressing clones, and they are a bit lower in the percent of MYC positive cells, but not significantly lower, to be honest. But they have no effect, even if the MYC levels are lowered a bit, because of, well, it's an amazing overexpression. The effect on proliferation is negligible. So even if one seems a bit lower, and indeed, in some of the replicas it is a bit lower, the proliferation is really not affected.
Thank you. I think this one's a good one for, I get the opinion of both Kristen and Akis here. Will spatial biology play a crucial role in the present paradigm? I think sounds like for studying the mechanisms of this disease in addition to high C.
Kristen, do you want to go first?
Sure. I think spatial biology is a really exciting area, and I think having that additional layer of information to add on to the 3D genomics is definitely a direction that is very exciting, and we would want to move into that space. I think it's not something that I've looked at personally in any of my stuff now, but it would be very exciting to kind of move in that direction in the future.
Yeah, one could imagine a method, where you image things in 2D on the tissue, and then you select the little bits that you want and you do a high C on them. Yeah, that'd be absolutely great. I'm sure someone that is, savvy enough will develop that pretty soon. The problem that I currently have with spatial transcriptomics, proteomics, is that the resolution does not match, for example, single cell resolution, right? It's still a few cells. So depending on the cancer specimen that you're handling, it can be very informative or it can be a bit blurry. These, of course, will get far better with time. So I can foresee this happening pretty soon, actually. I should, give a short mention here that there is a method that's called GAM, and Kristen definitely knows about it, which is a sort of high C alternative where you can look at the slide first and then check the nuclei you want to work with. So it has a spatial component to it. It's technically, I understand, there's a difficult method generally to establish in a lab, but it definitely seems to be in this direction of combining the spatial with 3D afterwards.
Got it. And it's sort of exciting because there's so many questions, and so I just wanted to say, if we can't get through them all, we'll definitely follow up and try to provide answers to some of these questions after the webinar. The next question is, in the cases in which you had multiple samples from the same patient, in particular the one with peripheral and central tumor sample, did you ever see an SV that caused a neoloop only in one of the two samples? Could the formation of these loops also depend on the tumor context?
So absolutely, we do see differences, and we see more than we expected. So the periphery and the central, are not really that similar. They are similar. They're the most similar in our cohort, but not really that similar. And they do have neoloops that are specific to one or the other. In the few tens, but they do have them. And these have been grown out. The tumor came out the same moment, right? The stem cells grew out in parallel in the same type of medium. So it's not just a technicality where you're subselecting different cells. There's clearly the periphery where perhaps cells are more migratory. It has some sort of different properties, functional properties, than the center of these tumor, apparently.
Awesome. Thank you. Next question, and I think we'll do this and one more, then we can, I think we'll have to close in respect out of Dr. Akis's time. And so the next question here is an incredible figure of high C displaying the true nature of loss of genomic homeostasis. However, is it homogeneous among all tumor cells? And I think this is referring to some of your slides, Kristen.
Yeah, I think a lot of our tumor samples are very heterogeneous. What we are seeing in a lot of those heat maps is kind of just a summary of the entire sample. We'd want to maybe get more into single cell or some of the spatial things that we were talking about to kind of really dig into that question a bit more. But yeah, I think the general answer, maybe Akis, if you have more insight on this, is it like
I actually wanted
heterogeneous?
To ask you, I mean, ours are incredibly heterogeneous, but what I wanted to ask actually is, I've always had this impression that, of course, the different types of signals don't have the same intensity, which you could interpret as a spectrum of, how many alleles or cells in the population do they actually occur to. Yeah, but I have not been able to come with an idea of how would you actually quantify that, right? So you have to have a range. What are we talking about?
Yeah. So I don't have a really, we've noticed that pattern as well, for sure, that there are some very strong signals that we can see in certain samples that we're like, oh yeah, this is probably like in 100% of the cells, like 100% allele frequency. It's really strong, really bright. And then there's other things that are just, they're faint, but they're very noticeable. They're very clear. We know that they're real. But as far as like putting a scale on that sort of subjective view of the signal strength that we're seeing, yeah, that's not something that we've really, like, we don't have numbers for that, but there's certainly some things that we can definitely say are stronger or not, and it's probably tied to either the frequency of cells that they're present in or the allele frequency in general.
I mean, one nice test would be to have an idea of what your lowest detection threshold is for, an event, for an amplification or a translocation, right? And one could do this in silico, maybe, right? You can mix two different samples and dilute it down and say, at which point do I stop seeing that? It's difficult, but I think it's worth doing at some point.
Yeah, we've done a little bit on that for some very strong translocations. I know we can go down really strong detection at like 1% or even lower if we're doing a targeted approach.
That is actually very encouraging, right? If you can see that sort of the 1% mixing, then this is pretty great.
Yeah, we know that we can detect things at pretty low frequency, at least in the in silico tests that I've done. Yeah. So that's part of what makes it hard for me to like put a scale on what allele frequency corresponds to what signal strength, because we do have really good detection limits. So.
Yeah, true.
All right, I was going to do one more question out of respect for your time. So the next question is, what is false positive result? It sounds like this is more about the question about like, are there false positives in the data and yeah.
I would assume that at least in our data, I would assume there's more false negatives. So I'm pretty sure that there are enhancer promoter loops that we're not capturing, despite how potent the kit is. And I'm not just saying it. We've seen that the Arima Genomics kit is very good for this. But I'm pretty sure there are enhancer promoter contacts that we're not capturing because of frequency and of contact and so on and so forth, or distance or whatever. And I'm pretty sure that there are structural variants that we're not capturing for reasons of frequency and resolution, because of course there'll be very small ones, and our resolution is really not less than 10 kb, probably 15 or 20. So a false positive result would be slightly more difficult to get, right? Because these are really strong signals that signify structural variants. And I don't know if Kristen has a different opinion on this, but I would assume that the false positives are far fewer than the false negatives. And if anything, okay, maybe we have a false positive deletion, small deletion, because we didn't map very well in that region, but that's probably about it.
All right. Well, hey, thank you, Kees and Kristen, for taking this time to share your knowledge and share your research and experience with Hi-C. And again, thank you to our audience. This was really a lot of questions and a diverse amount of questions. It was a great presentation. And for the questions that did not get addressed or answered, we will follow up with you, and we'll try to get back to you with the answers as soon as possible after the webinar. So again, thank you everybody for attending. This webinar again is recorded, and we will share the link with everyone involved. And please feel free to share it with friends and colleagues that might be interested. And with that,
thank you very much.
we'll close off the webinar. Thank you.
Yeah. Bye, everyone.